Hey everyone, welcome back. In this video, we'll run our first NF Core pipeline using Nextflow on our cloud server. Let's get started. So very quickly, what is NF Core? NF Core is a collection of standardized, best practice bioinformatic pipelines written in Nextflow. They're completely free and open source, actively maintained, new releases are made regularly, and they're widely used in both academia and industry. In this video series, we'll be using the NF Core RNA-seq pipeline, which takes raw FASTQ files and a reference genome as input and produces things like alignments and gene counts as output. And before using real data, it's common practice to run these NF Core pipelines using a small test data set to ensure everything runs smoothly. We don't want to run the pipeline using a big data set only for it to fail halfway because of some unexpected error. So let's do that first. So here's the command we're going to use. We use the run command to tell Nextflow which pipeline we'll use, in this case the NF Core RNA-seq pipeline. We use the profile command to tell Nextflow that we want to run the pipeline inside of a Docker container using the built-in test dataset. And the outdir parameter to tell Nextflow where to put the results. I also use these skip parameters to tell Nextflow which parts of the pipeline I don't want to run. And this is a great feature, especially for cloud computing, because it allows us to reduce the runtime of the pipeline, and this is going to save us money. When we run this command, Nextflow will download the pipeline from GitHub and execute each step of the workflow inside of an isolated environment using Docker. And you'll see all of the different steps of the pipeline running here, and if anything fails, it'll show up in red. The pipeline will take a few minutes to run through the test data, so let's come back when it's done. And if everything completes successfully, as shown here, you'll see a green completed message at the end, including information about when the pipeline completed, how much time it took, and the number of CPU hours. And one of the things that I want you to take note of here is that we just ran a full RNA-seq analysis without having to install a single bioinformatics tool. Docker and Nextflow handled everything, and that's why these tools are so powerful. Lastly, let's quickly take a look at the outputs that were produced. So inside the output directory, we'll find BAM files, which contain the aligned reads, count matrices, including gene and transcript counts, and QC reports, so pipeline summaries and information about data quality. So even with test data, this mirrors what a real analysis will look like and what we can expect to get when we run the pipeline on real data. Now we know the pipeline runs successfully on our cloud server. In the next video, we'll upload real sequencing data from my hard drive and run this pipeline on a real data set. I'll see you there.